Done. Yes. So, welcome to my first lecture, where we are mainly going to see how this topic of genome editing nucleated from the findings of Nobel laureates. As we are aware that all known lives like bacteria, archaea, or eukaryotes like plants, fungi, and animals, they all are connected by DNA. And the four-letter code here, ATGC, provide the code of life. Now, when these four-letter codes are strung together, they form a unique blueprint for each organism. And in mammals, billions of letters come together to encode more than 20,000 genes, as can be seen here. And about not only the coding genes, but there are non-coding elements and complex regulatory pathways we are only starting to understand. Now, the first human genome was sequenced in 2001, and since then, millions and more have followed. Now, the question remains that how this sequence information governs the cellular, tissue, and even full organism level function is which is completely unknown, or we are slowly trying to understand this, that all what these all codes or gene sequences mean with respect to the cells, with respect to tissue, and in full organisms. Now, to understand more, we need tools. Like engineers have many tools, as shown here, we biologists also need tools to reverse engineer cells and organisms, which would allow us to modify genes and study their functions, thus providing a pathway to understand the genetic circuits of life and what is the genetic circuits that drive the life. And indeed, we have now forward and reverse genetic approaches which allow perturbations to be made in DNA, RNA, or proteins in order to understand how the cellular functions are affected. Therefore, apart from these tools, we need precise tools to manipulate DNA at the specific sites which are required to enable these functional biology kind of experiments. And indeed, if we look today, we have different types of tools and strategies that have been developed starting from meganucleases, zinc finger nucleases, transcription like nucleases, and much talked about CRISPR-Cas9 systems. So before talking about all these systems, let's see how the field itself originated. Now, the very first tool to manipulate any nucleic acid in any cell was recombinant DNA technology that was developed in 1970. To, and this is a technology where you can express any kind of protein with the help of vector, appropriate vector, and when you include into the cells, it will express your protein of interest. And the first person to construct a recombinant DNA technology was Mr. Paulberg. He had ligated two DNA pieces, one coming from E. coli gene and another from SV40 phage, for which he had received Nobel Prize in year 1980. Now, what was the motive behind his work? He was very much excited about the recent discoveries made at his own time. First, genes were recently the genes were recently described as one gene, one protein kind of theory from Professor Tatum. And in the second place, from the Institute of Scripps, it was shown that SV40 was able to transform mammalian cells, and transformed mammalian cells were behaving like cancerous cells. So what he was convinced from the start, that we need some kind of tool to study isolated genes in vivo condition. And for this reason, he wanted to use SV40 as a tool to transfect his protein of interest or gene of interest into mammalian cells and express them in mammalian cells. So this was the basis of his work. With this basis in his mind, he started his work. What he did, he used SV40 vector as shown here. This is a small vector around 5 kb gene, and he also selected E. coli vector or E. coli plasmid containing 10 kb gene of galactose. So this then he started his work. When he started his work, this restriction enzymes were not known. It appeared only couple of years after he started his work. So for cutting off any gene, he realized he relied mostly on the every macloid experiments, where they had demonstrated that a viral gene in its lytic phase, they are very long and concatenated. So at the end of this lytic gene, they actually circularize and they form circular genes. So this is circularized based on their complementarity expression of not expression but complementarity nucleic acids at its complementarity 5 prime and 3 prime ends. So based on this knowledge, he started his work. He linearized first his plasmids and sp40 with the help of endonucleases, and especially with the help of 5 prime to 3 prime exonucleases activity. He chopped off the 5 prime sites from this direction as well as in this direction, both for sp40 as well as for E. coli gene. Then with the help of terminal transferases, he added complementarity at the 3 prime end. This terminal transferases activates only at the 3 prime ends of the thing. So here he added AAA and here he added TTT. He annealed these two together so that complementarity is forming base pairs, and to prepare the recombinant in the end, he used different kinds of enzyme that are used for repair enzyme. For example, DNA ligases, DNA polymerases in the presence of all types of DNTPs, and exonucleases, which are the repair enzyme. So based on this work, he was able to successfully carry out construction of recombinant DNA, where he ligated SV40 with the E. coli DNA. Now, as I said, his main motto was to construct hybrid genome and then to transfect into mammalian cells. So here he wanted to transfect into mammalian cells to see if galactose can be expressed, and also he wanted to express in bacteria to see if SV40 can be propagated in E. coli cells. So when he presented his work towards the scientific community, they said that the whole thing is too risky because SV40 is a phage and has infectious property. If it is leaked from the laboratory, then this bacteria or cells would carry this phage and can infect any other organisms. Nevertheless, the second caveat in his experiment, he realized that he had not included any origin of replication in his construct. So, after this experiment, he did not proceeded further for the transfection into the mammalian cells or transformation in bacterial cells. But few years later, restriction enzyme had come into the picture, and this was and many people had started using restriction enzyme. So he again started performing his experiment, and by this time he was overcoming these problems. For example, first he included origin of replication in his construct. Second, by this time he actually learned how to propagate SV40, and here he he had learned that how to actually minimize the transfection property. For example, if we see, this is the construct of SV40 where it expresses early genes and it expresses late genes, as can be seen here. So now if you use this SV40 to generate your protein of interest, that means either you are going to cleave this end or you are going to cleave anywhere at this end. So if you cleave this end of SV40 to insert your protein of interest, for example here, that means you are deactivating early gene. So in case you are deactivating your early gene, then he actually came up with a system where he has another SV40 vector, and the early gene is expressed in this SV40, whereas the late gene is defective in this case. So this is known as helper cells. And since the late gene expression is coming from this SV40, so an early expression is coming from this system. So this kind of system is currently known as a complementation assays. So he generated this, and in case if you are inserting your protein of interest in the late gene, that means you construct a helper cell where the late gene is expressed correctly, but you do not have a early gene expression. So this is mutated. So that means there is no early gene expression. So with this kind of system, he actually started his work and he overcame the other problems in this case. So the risk element he actually avoided, and again he transfected his protein of interest in mammalian cells and checked for the expressions. But to his surprise, whatever gene he was trying to incorporate, he was not able to see any expressions. There was no expression. And the reason he came later to know that by this time only the all scientific community were knowing that all the genes are similar like bacterial genes. So in bacterial genes there are no introns, whereas in eukaryotes we have introns and exonic sequences. So he was taking genes from eukaryotic systems and inserting into this recombinants and expecting this to be expressed in mammalian system, which did not work. But after knowing the expression, or after knowing the mechanism of splicing, it again became easy for him, and then he only took cDNA or the open reading frame of the eukaryotic genes and inserted into the SV40 vector, and after that his fortune changed. And the first gene he tried in his mammalian system was beta globin coming from the rabbit. So he expressed beta globin gene and to his surprise, the expression was very good and he could find out that the globin gene was expressed. And after this, lot of different genes coming from various systems, for example, yeast, mouse, or plants were successfully expressed following his experiments. So this was his trial, and that is how the whole field started. The expression of genes or editing of genes. This is how the whole system started. Actually, how to convert one gene into another system and how to express it, how to analyze it. So another big discovery happened and took the whole field of editing forward. And basically, it gave a lot of boost to this editing field, and this was the discovery of restriction enzyme by this three gentlemen, namely Werner Arber, Daniel Nathans, and Hamilton Smith. For that, they received Nobel Prize in physiology or medicine in year 1979. So apart from this discovery what they made, and there are a lot of different papers available based on their work, but what I found I want to share here with you, that it's a 10-year-old girl writing who wanted to know why his father Werner Arber was receiving Nobel Prize. So according to her, there is a king. For example, I will just explain what she thinks about restriction enzyme. There is a king and there is a servant, and the relationship between one to another, there is a relationship between one colonies to the another colonies, and king here is your DNA, which is long and skinny, and the servant is your enzyme. This enzyme only cuts the foreign DNA. If there is any foreign DNA who is trying to invade this colony, this enzyme will cut this foreign DNA into pieces. So this enzyme will not harm its own king. It will not chop off its own king, but this servant will only look after the secrets of another king invading into the system. So let's see what she writes, and I quote here. Especially when I come to the laboratory of my father, I usually see some plates lying on the tables. These plates contains bacteria which remind me of a city with inhabitants. In each bacterium, there is a king which is very long and skinny. But the king has many servants. These servants are thick, short, and like balls. My father calls the king DNA and servants enzyme. The king is like a book in which everything is noted on the work to be done by the servants. For us humans, these instructions of king are a mystery. My father has discovered a servant who serves as a pair of scissors. If a foreign king invades a bacterium, this servant can cut him into small fragments. But he does not do any harm to his own king. Clever people use the servants with scissors to find out the secrets of the king. So to do so, they collect many servants with scissors and put them onto a king so that the king is cut into small pieces. With the resulting little pieces, it is much easier to investigate the secrets. And for this reason, my father received the Nobel Prize for the discovery of servants with the scissor. Now let's see how restriction enzymes are classified. So after discovery, these were classified into various types. And here in this lecture, we will see the three types of restriction enzymes. These were classified based on the domain structure, cofactor requirements, length and symmetry of recognition sequences, position of cleavage, and their mode of action. And out of all three, basically the most important belong to the type 2 enzyme, that is type 2 restriction enzyme. And currently we have more than 4,000 type 2 restriction enzyme that are used in molecular biology experiments. Now it is interesting to know that the ubiquity and the variety of type 2 enzyme in different species is thought to result to come from a common evolutionary basically need for a strong defense mechanism. For example, here, if you see, you have defense mechanism against phage and other invading DNA. So restriction enzyme chop off the foreign DNA as can be seen here. So it is reasonable to question that bacterium own DNA, how its own DNA is protected. So it was found that restriction enzyme of all three types or all types have cognate DNA methylases enzyme that recognizes same DNA sequences as endonucleases. And this methyl transferases catalyzes the transfer of methyl group to a specific base within the recognition site, as can be seen here. Now there are various nucleotides that can be methylated. For example, you have cytosine that can be methylated into 4-methyl cytosine or 5-methyl cytosine or 5-hydroxymethyl cytosine. So apart from cytosine, even adenine can be methylated at position number 6. Thus the methylated, when these bases are methylated, they are not able to cut by the restriction enzymes. So this is the basis that their own DNA is protected against restriction enzyme. Now on bacterial gene, if you see, the restriction enzyme and its cognate methylases are mostly tightly linked. For example, if you see there is one restriction enzyme going from here, so there might be immediately next to next to it there might be methylases gene also, not every time in the same orientation, but it can be of a different orientation. But this depicts that there is a tight linkage between methylases and restriction enzyme nucleases, and this tight linkage is to decrease the chance of separating these two genes during the process of conjugation or the process of recombination events, so that they go together from one species to another species. Now there are few things, important things about restriction enzymes, and these are: first, recognition domain or recognition site. Second is your cleavage site or endonuclease site. And apart from this, you have third thing is methylation site or methylase enzyme. So apart from these three important factors which we should know, there are cofactors involved, and these cofactors are written here: SAM, SAM, S-adenosyl methionine, or ATP, or sometimes magnesium. Most of the time magnesium is involved, or some other kinds of yeah. So all these complexes, for example, all this either recognition site, cleavage site, or methylases domain can be present in either one complex having all the uh, necessary items, for example, endonucleases activity, cleavage activity, or recognition activity inside one complex of a thing, or they can have in independently or in terms of dimers. We'll see. So in type 1 enzyme, this whole enzyme, type 1 enzyme is kind of a complex where the three different subunits come together. For example, you have endonucleases, you have methyl transferases, and you have recognition, for example, for specificity. See, so all these are together in one complex, and this is how they function. And as a cofactor, they have S-adenosyl methionine and ATP for translocation activity. Apart from that, they cut asymmetric DNA. That means they do not have any symmetry. And the cleavage site is far apart. That means it cleaves while it is going on the translocation of the DNA. So we will see a video of type 1 enzyme.
Type 1 restriction enzymes
Are you able to hear?
consist of three.
Ayushi?
Yes, ma'am.
Okay. Can we?
Type 1 restriction enzymes consist of three proteins that function as a single protein complex. The complex typically consists of a specificity protein, two methyltransferase proteins, and two restriction endonuclease proteins. The type 1 restriction modification systems recognize a specific bipartite sequence with the half sites separated by 5 to 8 nonspecific nucleotides They do not, however, produce a predictable cleavage pattern. Once the protein complex binds the DNA, it acts as a molecular motor, using ATP to translocate thousands of bases along the DNA molecule until it encounters a second complex, at which point it creates a double-stranded break. Because cleavage occurs after translocation, the cleavage site is not fixed. As a consequence, there is no distinct cleavage pattern. In addition to the requirement for ATP, type 1 restriction systems also require S-adenosylmethionine and magnesium ions. At this time, there are no type 1 restriction enzymes that are commercially available.
Coming to type 2 enzyme, which is a vast in number, as I said already, more than 3,500 or 4,000 enzymes in this class is known. So they can act as a monomer, they can also act as a dimer or tetramer. So if they act as a monomer, they recognize smaller sequences, 4 base pair. They can recognize 6 to 8 base pair if they act as a dimer, and they can act as a tetramer. In this case, they recognize more than 2 recognition site. So the recognition and cleavage site is within the one dimer. So it has recognition site as well as it has cleavage site. So that means it cleaves within the recognition site. And the methylation enzyme is totally away or apart from this complex or homodimer forming here. There's another enzyme called methylase, which actually recognizes the same recognition sequences for the addition of methylation. So apart from this, I want to show you an example where the enzyme has evolved different mechanism for binding and cleaving. For example, here you have BAMH1 and BGL1, which recognizes 6 base pair targets with 4 central bases as G8TC in common. So G8TC here. Now these two enzymes use completely different arrangements of amino acid to contact the central bases. That means they have differences in their active site, suggesting their specificity could not be easily changed just by point mutations. For example, if you change here G8TC to GA8CC, that means you have a point mutation at the place of T to A. And this will lead into not the identification of BAMH1 at this site, because BAMH1 will not recognize the mutated site and hence you will not have the cleavage at this position. But this will have a deleterious effect or lethal consequences, mainly because the cognate partner of methylases will not be able to recognize this mutated sequences and hence it will not be able to protect this sequence. Therefore, just having one mutation you will not allow it to change the whole system. So it is very difficult to mutate any restriction enzyme, leading it to different specificity. For example, you cannot easily take a mutation from one site to another site, and that has the consequences that your methylation enzyme will not be protecting your this new target sequences. So this means that the whole enzyme or the active site of the BAMH1 and active site of BGL2 here, they have completely different way of approaching the central basis G8TC. Now let's see a video on type 2 restriction enzyme.
Type 2 restriction enzymes are the kind used for most molecular biology applications, such as gene cloning, DNA fragmentation, and analysis. They cleave DNA at fixed positions with respect to their recognition sequences. Over 3,500 type II enzymes have been characterized, recognizing over 350 different DNA sequences. Restriction enzymes are named for the microorganism from which they were originally purified. For example, HinD3 was the third enzyme found in Haemophilus influenzae, serotype D. Type II restriction enzymes vary widely in size, amino acid sequence, domain, organization, and subunit composition, cofactor requirements, and modes of action. They are loosely grouped into subtypes based on their enzymatic properties. The four most important subtypes are type 2P, 2S, 2C, and 2T. Type 2P enzymes account for over 90% of the enzymes used in molecular biology. They recognize symmetric or palindromic sequences, 4 to 8 base pairs in length, and generally cleave within that sequence. The subunit composition of type 2P enzymes depends on the length of the enzyme's recognition sequence. Enzymes that recognize short, 4 base pair sequences act as monomers, comprising single protein chains, while enzymes that recognize longer, 6 to 8 base pair sequences typically act as homodimers, comprising 2 identical protein chains. Still other type 2P enzymes act as dimers of dimers, or homotetramers. These latter bind to and cleave 2 or more recognition sequences at once. Upon cleavage, some type 2P enzymes leave single-stranded overhangs, while others leave blunt ends.
Now coming to type 3, they are again a complex with two subunits as it's shown here. There the subunits are endonucleases are present and methylase recognition is present. For example, they also need S-adenosyl methionine as well as ATP for translocation and magnesium for the cleavage enhancement. For example, here again the type 3 restriction enzyme cleaves DNA by long range interaction between the recognition sites arranged in the head to head or tail to tail configuration. So it is depending that how these two sites are complex, and like type 1, this type 3 restriction enzyme are also complex, and these complexes make it a little harder for the scientists to purify individual subunits from this complex and contain or reconstitute to understand that how they work and what is the mode of cleavage for these enzyme, because type 1 and type 3 they both actually um, move or translocate and then only they cut, so their cutting site is not fixed most of the time. And we will see next the video on type 3 restriction enzyme.
Type 3 restriction enzymes are infrequently used in molecular biology as they have few relevant applications. Therefore, they are less well characterized than type 2 enzymes. While the mechanism of type 3 restriction modification systems is uncertain, the enzymes themselves consist of 2 proteins that function as a single protein complex: the methyltransferase, or M protein, which contains the specificity domain, and the restriction endonuclease, or R protein. Type 3 enzymes recognize a 5 to 6 base, non-palindromic sequence and require 2 inversely oriented recognition sites for cleavage. Once bound, the enzyme slides along the DNA and upon encountering a second complex, cleaves downstream of the recognition site, typically 25 to 28 bases away. Type 3 enzymes require ATP, S-adenosylmethionine, and magnesium ions.
So with this, I'm already at the end of my today's lecture. So in this lecture, we saw a brief historical start of genome editing, It started with the work of Mr. Paul Berg. So how he mutated nucleic acid, he joined two genes together, and then he created a vector to transfect a gene of interest into mammalian cells. Then the whole system or the whole field was enhanced by the discovery and classification of restriction enzyme. Today we also saw type 1, type 2 and type 3 restriction enzyme. We also saw an interesting perspective of a 10-year-old girl on restriction enzyme. In her mind, the restriction enzyme works as a pair of scissors to cut for an invading DNA. Now, this whole restriction enzyme and the basis that how the whole field started will form the framework for subsequent lectures, or in future lectures we'll talk more about restriction enzyme, and especially we'll talk more on type 2 S restriction enzyme, and then we will go further on type 2 S, and then typical example of FOC1 enzyme we will study in our subsequent lecture. So with this, I thank you for this class. Okay.